Well, I'm here with Dr. Robert W. Malone. He's an MD, and he is known as the inventor of the discoverer of the vitro and in vivo RNA transfection and the inventor of mRNA vaccines. And we welcome him to the program now. How you doing, Dr. Malone?
I'm doing okay, David. I hope you're the same. I hear you're a little under the weather a bit.
Yeah, but we're gonna just have a great discussion anyway. So you have been on the radar for a lot of people because of your criticism of the public health authorities' perspective and approach to the pandemic. And someone who, like yourself, who has created the mRNA technology behind these vaccines, has been someone that people want to pay attention to. We're told to follow the science, and so if you're the scientist that helped create the technology at the core of these products, it's good to follow what you have to say on the matter.
Well, that's kind, and I think that's a good, accurate description, the core technology at the heart of these products. I think that was well said. I'm a lot more than that, though. I've been involved in outbreak response, both on the new technology side and on the implementation side since the 1983. I've been through way too many of these and been through a bunch of them since I moved over to support more of the DoD side right after the anthrax attacks. And I've been doing that pretty intensively ever since. So I understand the government pretty well. I interface with a lot of govies and understand their systems and processes. And I've lived multiple outbreaks, being at the tip of the spear in response. And so I think I'm reasonably well qualified to help folks navigate the Byzantine world of DC and CDC and the technology.
So you said you've been involved with outbreaks. What does that mean?
Well, for instance, just recently, the last few years, H1N1, Zika, Ebola, and now this one goes back to the various earliest days of AIDS. I've supported BARDA, a big HHS contractor, in developing new flu vaccine technology. I was right at the tip of the spear in bringing forth the Ebola vaccine that we now call the Merck vaccine, and I was the one that brought Merck into that deal. So I've really been kind of boots on the ground. I usually am below the radar. My customer, Department of Defense, really doesn't like me being out here and being outspoken in an official sense, and so I don't talk about them or their policies or what we're doing because I'm not authorized to speak on the behalf of the Department of Defense. But my colleagues are largely really supportive, but behind the scenes and quiet because they don't have the authority to speak out.
So what makes you have the option to speak out, and why did you speak out now?
Fortunately, right now I'm working just for subcontractors, and they are comfortable with how I struck a balance here. And I'm not speaking about their products, so I really don't want to talk too much about what we're doing together with our drug repurposing work, and that's the telecon that I'll be on, the Zoom that I'll be on it too. But they're okay with me speaking out in this context. And on the more direct DoD customer, I'm just really careful to not entangle them in things, and they recognize that I'm doing that.
So when you, as an MD, did you see patients, or have you been a research scientist? Did you ever work in a university? What's your background in that?
I was a university researcher and taught pathology, immunology, etc. for about a decade between 1991 and 2000. And then I switched over to focusing on advanced development. So that's the clinical research, etc. after the outbreak, the anthrax attacks. So I saw patients when I was in training pretty intensively at Northwestern University. And then the patients that I saw as a pathologist in training at UC Davis, unfortunately, were not very active. You'll appreciate. So I'm not a primary patient care person. I was for quite a while a discovery research scientist and also an entrepreneur starting new companies. And then after the anthrax attacks, I've switched over to focusing on more bigger pictures of assembling teams, managing teams, building large contracts, and managing them on behalf of my customers to solve, various complicated problems involving predominantly biodefense and medical countermeasure development. So all these various flu vaccines, et cetera, that we've seen over the years.
So when you talk about being the inventor of the mRNA technology, what does that mean? What does that look like? Describe what you discovered and how this was involved with the products that we have.
So
now.
This is at the very, very early in my scientific career when I was still a graduate student trainee at the Salk Institute in UC San Diego, and then following a short gig at a biotech startup called Vical. I was there. I had already finished the first 2 years of my medical school, which are the preclinical years, and was doing graduate work, had chosen to focus on retroviruses and the use of retroviruses for gene therapy purposes. And I'd sought out the best laboratories in the world to do that and managed to get myself into one that was a postdoc lab. So no other graduate students. I was the only graduate student at Inder Verma's lab at the Salk Institute between 1983 and 1989. I'm sorry, 1987 and 1989. And so there I was focused on some fundamental questions having to do with RNA structure and biology as it relates to retrovirus packaging. And in order to ask those questions, I had to develop some technology platforms. I was also involved in working with a postdoc that was doing some fundamental retroviral gene therapy work, and that comes into the story also. So it was a series of about 5 discoveries that stacked one on top of the other. One of the key ones that relates to this area was that my kind of postdoc mentor, Dan St. Louis, was working on a model where he would use retroviruses to put a gene, a transgene, into a mouse cell and then implant those mouse cells as a pellet into a mouse and then follow the expression of the protein that could be therapeutic potentially. And after about 3 weeks, that was lost. The protein expression was lost. And I was the one that figured out that the reason it was being stopped was because the immune response against the cells that were making that protein. So that was kind of a crucial moment because it meant that the whole focus of my graduate work and the career that I'd envisioned as a gene therapist was suddenly a problem because it meant there's a fundamental flaw in the gene therapy concept, which is that these foreign genes were going to provoke really strong immune responses. And it was a make lemonade out of lemons moment. I'd already had a strong background in vaccinology from the lab that I had come from at UC Davis that was involved in building candidate vaccines for HIV and the monkey model of HIV called SIV. So I was pretty well entrenched in vaccinology, also in the virology and vaccinology of influenza, but then realized that this could be an application for the gene therapy tech. Then on top of that, there were a series of, and that, by the way, that insight gave rise indirectly both to this RNA vaccine technology, to DNA vaccine technology, and to the use of adenoviral vectors for that purpose. The senior postdoc in the lab founded Crucell when he left, came to me a few years later and said he was going to pivot Crucell, an adenoviral vector company, from gene therapy to vaccines, and thanked me for my ideas in that. That company, Crucell, was sold to Johnson & Johnson, and that's the technology platform that gives us the J&J vaccine. The RNA vaccine story, as I said, I was working with RNA, had for years,
right.
And I had to develop a system to allow me to make large quantities of RNA, be able to detect whether or not they got into cells efficiently, and come up with, I tried a bunch of different methods to try to get that RNA into cells until a senior scientist at the Salk, Tony Hunter, suggested to me a new technology that had just been reported at a company called Syntex in the Bay Area, which was the use of cationic lipids, positively charged fats, that self-assemble with DNA or RNA to form nanoparticles, nanomolecular complexes. And Syntex had been frustrated in trying to get RNA to work. They'd made the DNA work. So we set up a collaboration. They shipped me their reagent. I added it to the technology and the RNAs that I built. And that's what suddenly opened everything up. And soon thereafter, I was delivering RNA efficiently into every cell type I could test that I gathered from the salt, as well as frog embryos. And then that set up a whole patent fight and a whole bunch of politics between the Salk and UC San Diego that left me in a position where I just psychologically couldn't stay in that environment anymore. And I left and joined a little startup company that had already employed one of my collaborators from Syntex. It was called Vical. And Vical set me up at the Skunk Works. And long story short, within a short period of time, I was only there 4 months. I brought all my reagents, technology, protocols, etc., and the disclosures of initial patent disclosures that I'd filed with the Salk, and started testing the use of these molecules in mice, together with a researcher from the University of Wisconsin that had used to be at UC San Diego that I'd known, named John Wolfe. And a cascade of events led us to find that in the mice, you didn't even need to have the positively charged fats. You could just inject the naked RNA or naked DNA and get both a expression and an antibody response, and then subsequently a cellular response. John Wolfe was a pediatrician focused on classic gene therapy, and he thought that this would be good for curing muscular dystrophy. I knew the levels of protein that were being produced was really small and would never be useful for gene therapy purposes, but that we could use it to generate immune responses. So that ended up with 9 patents that issued, a long series of discussions with lawyers, and we pretty much envisioned the entire scope of what now is considered to be mRNA as a drug and mRNA vaccines, put them all in patents, had the data showing that we reduced that to practice, and the patents issued and were used by Vical, and then Merck bought those for $9 million and tried to develop products. Both Merck and Vical kept everybody else out of the field for the 20-year lifespan of the patents, and it was only when they expired that suddenly you had kind of this groundswell of new companies popping up. So that's why there's a 20-year delay.
So when you look at these current vaccine products, how are they connected to the technology you're describing in those patents?
So they all use the same basic steps. They use the same manufacturing process that I scaled up, manufacturing and purification, but they've scaled it up even more. Before that, no one had ever really done large-scale mRNA synthesis and published it. There wasn't a reason to do it. It didn't make sense. They were making small amounts of RNA for small reactions in a few microliters. So making a milligram or more of RNA had just not been done before. And they're still using that process, but scaled up. They're still using the same structure of the RNA. I had to define various molecular elements that would make it work. And they still use that. They've improved on it some. And they still use the condensation of the RNA into a nanoparticle using a cationic lipid, but it's a different cationic lipid. It's one that has a variable charge rather than always being positive. It's positive in some contexts and then becomes less positive or neutral based on pH as it goes through the cell fusion process. That was done by Peter Collis and his group at the University of British Columbia. That was a key advance that led to this current technology. The stuff that we were working with and the applications back then were blocking RNAs, antisense RNAs. I've also published on the use of ribozymes. These are RNAs that cut like scissors, other RNAs, molecular cutting, and antisense. There's a bunch of other technology applications. And in the vaccine space, our focus was on HIV and influenza. The influenza led to a big paper that Merck published in Science, but never led to a product. And the disclosures that I'd originally filed around a method for universal influenza vaccination never got pursued. What really happened is that Merck and Vical decided that it was just too darn hard to make RNA to do what I was doing. And I don't think that they had anybody in the lab after I left that was very good at it. So they decided that was just going to be too troublesome, too expensive, and they could just work with DNA. And unfortunately, all the people that have tried to develop DNA vaccines have failed, including Merck and Vical. But it was this original idea of using RNA that's turned out to be successful for a variety of different reasons. Have I answered your question?
Yeah. So what is your overall assessment of these products that have been rolled out by Pfizer and Moderna and Johnson & Johnson?
So the J&J tech, and there's also the Sanofi, Aventis, Cambridge, AstraZeneca virus vector that is similar, that's used in Europe more. The adenovirus vectored ones are based on a gene delivery technology or gene therapy technology that's designed to produce high levels of protein for long periods of time. The RNA strategy is designed to produce lower levels of protein for just a short period of time, and then the RNA gets cut up. So that's a fundamental difference between the two. In all cases, what's happened is the FDA has applied the vaccine checklist for what they've required the pharmaceutical companies to do, and they haven't applied the gene therapy checklist. So as a consequence, there's a bunch of things that they might have done or they would normally do for a gene therapy strategy that haven't been done in characterizing these. And one of those key ones is that the pharmaceutical companies weren't required to characterize how much protein was being expressed, where, in what cells, and for how long. So as a consequence, with this kind of a gene therapy strategy with both the RNA and the advectors, what we're doing is making your cells into the factories that produce the RNA, the vaccine. And it's important to know how much vaccine is going into the patient. So if you take a traditional jab, that's very well characterized. We know exactly how much protein. But the FDA hasn't characterized that. They haven't characterized the reproductive toxicology, the genotoxicology, or a number of other things. So one of the things that we have here is a situation where all of these vaccines use a protein from the virus that was known to be highly immunogenic. But when they engineered them, they didn't know that this protein was also capable of causing problems, pathology, sickness in people. And so now we have the infamous spike being produced. And spike, we now know in the virus infection itself, causes a lot of the disease directly. And the spike that's being produced in the vaccines is very, very similar. It has two tiny little mutations put into it that cause it to move from a flexible conformation that can either bind its receptor or can inject the virus into a cell. That's how spike works. It is like a little pump that has both a binding part and then a pump part. And the mutations lock it into the binding conformation so that it's easier to make antibodies against the inside of that binding pocket. But it turns out also that the antibodies that are best are really on the edges of the binding pocket and at the junction between two of the three subunits that form this complex. And so that whole strategy in retrospect didn't make a whole lot of sense. And the logic of using an intact spike protein doesn't make sense in retrospect because we now know a lot more about the toxicity than we knew back then. So we're in a situation with the first generation type products that relied on kind of the low-hanging fruit, the easy pick. And it's turned out that that easy pick maybe wasn't so smart after all. And I think we're probably going to see second and third generation products. Now, that on the RNA side, with the adenovirus vectors, those have been used again and again and again in humans in a variety of different vaccines, including some of which are licensed. And we know what the problems are with them, but the problems that we're seeing here are different. Okay, so they're probably more related to what's the payload, the thing that the virus is putting into your cells and causing to be made, than it is with the basic technology platform. With the RNA vaccines, they have new chemicals that have been made in the laboratory. They're synthetic. And we know from basically a leaked Pfizer document that those fats, those new fats, accumulate in some parts of the body in a way that is kind of not readily understandable. It doesn't make sense. It seems to be some phenomena that we don't understand. And in particular, the thing that has got some folks worried about is that these lipids accumulate about 12% in the one study we have of those lipids that are injected end up accumulating in the ovary and not in the testis. So that raises the whole specter of reproductive toxicology. And the honest truth is we don't know whether they're safe or unsafe because we didn't do the animal studies and we haven't had enough time to figure out whether safe or not safe in the humans. Does that all make sense?
Yeah. What about the factor that these vaccines were designed for the original Wuhan strain, and now we've got many other Delta variants and all these other variants? Doesn't that make them ineffective at dealing with the original if the original doesn't exist?
So that's a hot topic right now, and there's a lot of debate, even among virologists and vaccinologists, about how important those mutations really are. It appears that, and the data is still coming out about Delta, there's a lot of different ways to interpret those data. But normally one would expect that in any situation, viruses are little evolutionary race cars. They produce huge numbers of offspring, and many of those offspring have mutations. And many of those mutant viruses are no good for anything. But some of them are able to slip through other protections that the body has, including an adaptive immune response. So what you've got is a situation, a natural situation that always occurs when a virus moves into a new population, that, which is the case here, no matter where the virus came from, it certainly has a new host in all of us. And as it spreads and replicates, it's being selected for changes in its characteristics, including the amount of virus that's produced when it infects you, and including its ability to evade any pre-existing immune responses you may have. So in the case of a vaccine that's only against one of the virus proteins and is only partially able to block the ability of the virus to infect or replicate or infect others, which is the honest case here. So the vaccines are pretty good at protecting against death and disease that for people that would be at high risk of death and disease. But they also don't particularly protect well against the infection. They were selected for protecting against death and disease. So we've got a situation where the virus is still infecting people that have vaccine and have those antibody responses. And it's expected that these viruses will evolve to escape that. Now, why does that matter? There's a particular problem with, so long, you could say, well, who cares so long as it's protecting from death and disease, right? So we all get our coronavirus infection, but we all get infected with circulating coronaviruses anyhow. There's beta coronaviruses that we call the common cold. So then the question is, who cares if it's still protecting us from death and disease? there's a particular problem with coronavirus vaccines that we've known about for decades, both in animal vaccines and in human vaccine development, that's pretty much compromised every prior coronavirus vaccine candidate in humans and most of the veterinary ones. And that is called antibody-dependent enhancement. And antibody-dependent enhancement is, if you think of an antibody as like a fork, and these are the tines, they're the parts that bind to an antigen, say a virus surface. The fork has a handle, like my wrist, and that also has a binding sequence, but what it does is it binds to certain cell types, immune cell types called monocytes and macrophage. And that's important because those cells use that to gobble up viruses and chew them up after they've been bound by antibodies. But antibodies that aren't good at blocking the ability of the virus to fuse with the cell and infect a cell, but are still good enough to bind it and allow it to bind to macrophage and monocytes, can cause those cells to get infected in a way that they don't normally get infected. And so you end up with a bunch of cells being infected that, by the way, move around in the body that normally wouldn't be infected. What do you see when that happens? Well, one of the things you see is a more explosive sudden onset disease. And another thing you see is much higher titers of virus in people because the virus is infecting other cells than it would normally infect. So this, in the case of dengue, which is the prototypic virus associated with antibody-dependent enhancement, it's got 4 different strains. And if you get infected by strain 1, it provides partial protection against strain 2, but not really good protection. And so if you then get infected by strain 2, you get a catastrophic disease called dengue hemorrhagic fever because you get this kind of explosive replication of the virus. So that's the problem with having a, half-baked immune response or a virus that's learned to escape a vaccine, particularly with the coronavirus.
If you don't have a full spectrum defense, the vaccine defense may accelerate the explosion, or
may actually make it worse.
Yeah.
Yep, may actually make it worse. And that's a known problem with coronaviruses. And there's some signs in the data coming from Israel that where you're seeing of those that are infected and going into the hospital and having severe disease, a large fraction of them are people that had been vaccinated before. And we know that this Delta variant is associated with higher replication in the body, whether or not you're vaccinated. So those are signs that we would be looking for. It doesn't prove that we're having antibody-dependent enhancement, but it's signs that it could be happening.
So in some cases, the vaccine may actually cause a more severe case of the COVID, or not? We're not sure yet.
Precisely, especially on the waning phase, the far side after vaccination. So vaccine responses come up really fast and then they taper. And what we know from the Israeli data, and the government has now confirmed, and Pfizer had alerted us about 2 or 3 weeks ago, is that the Pfizer vaccine is only being good for about 6 months, which is why they say they want boosters. Why would they need boosters? Well, because in that declining phase, there's a large window of time where you no longer have protection, but you have enough of these kind of crummy antibodies that you can still get things like antibody-dependent enhancement. And so there's a long window there where there could be something happening like I'm talking about during the declining phase. And we know that's a problem with dengue vaccines, and it could be part of what's going on with the Israeli data, where they had vaccinated everybody, with high efficiency very early on, and so people were more synchronized. Now they're about 6 months out, and they're seeing these cases coming through, including the breakthrough cases in the vaccine recipients. So it's still a murky picture. That's the problem with all of this, is that there's never perfect data in an outbreak. And if you want to clean all the data and make it look perfect and account for all the variables and the other confounding things, it takes about 4 to 6 months. And so if you only want to work with the perfect data, you're always going to be way behind the curve. So you kind of have to work with the raw data. And yes, everybody's right that's saying, well, those data are not perfect. You never have perfect data during an outbreak, and you have to try to figure out the ghost in the machine, the signal from the noise, by looking at trends that balance out all those other variables. And you look for trends in the data, like a sudden spike in deaths, for instance, or a sudden spike in hospitalizations. And we're seeing those kinds of things now. And the question is, what does it mean?
Now, what about the Biden administration and Fauci's argument that all the people who are getting sick from COVID and dying are in very low unvaccinated areas? And they're trying to say it's all the unvaccinated people who are getting this new Delta.
So there's a counter argument to that has to do with the seasonality. And there was surges previously in the Northeast where people are highly vaccinated, and then they were followed by surges in the South. And those surges didn't have a whole lot to do with vaccination status. So the logic that this is all happening in people that are the laggers, the slackers who haven't gotten their vaccine, and that's driving all the mutations, that's just false. The data are quite clear from other countries, and they're starting to become much more clear. Remember, here in the States, we're fairly early on with Delta. So there are reports coming out of, for instance, Florida, where there are a significant number of cases, a place near and dear to your heart. And I'm acknowledging that you live down there.
Yeah.
And there are nurses and others reporting that, in fact, a lot of the cases they're seeing are in previously vaccinated people. So I don't know what causes Tony to say what he says. He's got a long history of saying things and then flip-flopping. Like he said, oh, no, you can't say anything about the Pfizer vaccine. Most recently, he reprimanded Pfizer for saying we were going to need boosters 2 or 3 weeks ago. And then just a couple of days ago, the government comes out with a statement that we're going to have to revaccinate everybody that got Pfizer that's in the high-risk groups. So Tony has a habit of substituting his opinion for facts. I'll just leave it at that. That I haven't seen data, certainly from those countries that have much better reporting than we do, like Israel, that demonstrate what he is asserting, that the infections are occurring in the unvaccinated. They're in both the UK and Israel. They're clearly happening in both infected and vaccinated and unvaccinated. And there's some curiousness to how these infections are behaving in the people that have been vaccinated previously.
Now, what about natural infection immunity? Does that seem to last only 6 months or so too, like the other one, or does that have more long-term defense?
Not well characterized. What one would expect is that because the natural infection, there's a lot more virus antigens, not just spike, that it would be a lot harder to develop resistant mutants to the natural infection, because the breadth of immune response is going to be greater because people have experienced more antigens in the natural infection. That isn't always the case, because the virus has a lot of tricks that it uses to twist and turn how your body's immune system responds. But that seems to be the case in terms of the durability, this length, how long are you protected after you get infected? That's something that has to be determined scientifically. And unfortunately, the focus has all been on vaccine, not on what is the effect of protection afforded by the natural infection. So we just don't have the data.
So is it looking like we're going to have boosters indefinitely now, since these things are lasting only 6 months or so?
So there's an argument about that, and I've got to drop off soon,
Yeah.
just to let you know. But there's a case to be made that vaccination into a pandemic, into a widely spreading and replicating virus, kind of results in something like the nuclear war fight between the U.S. and the Soviet Union, where I come up with a bigger bomb and then you come up with a bigger bomb or more bombs, and we just go back and forth and back and forth. It's kind of that situation. If you keep trying to improve vaccines and vaccinate into a pandemic, is the virus will escape those, and then you'll put more selective pressure and it'll escape those, and it goes like that. So there's a good logic that it would make a lot more sense to vaccinate the people at high risk and let the others alone. Make sure they've got drugs. That's another part of the problem, is we're not investing in repurposed drugs or things that can be used as outpatients to treat people that might have COVID infection but aren't so sick that they have to go to the hospital. And that could be a better way to get out of this. And then we might gradually get towards a virus that is more like the regular circulating cold viruses or flu, just through the natural evolutionary process. But that remains to be seen. But there is a very good case to be made that vaccinating actively the whole population in the face of a pandemic is a bad idea.
Does that mean it might make more lethal viruses or that escape, or would it just be more virulent but not lethal because they lose some of the power?
So now you're in the coulda, shoulda, woulda land, and a lot of things can happen. What it could do is make for viruses that are much better at escaping vaccines. That would be a probable outcome. And we would really prefer to have vaccines for pediatric populations once we do reach herd immunity. Because what will happen once we do reach herd immunity is that gradually it's like filling up the glass with more water with the birth of new cohorts. And then the glass gets to a certain depth that the fish can live in it. If I'm using the metaphor, the goldfish can be put in the glass, not the glass that's this big, it won't live, but if it's a glass this big, it will live. If you could think of that as a metaphor for the virus, the goldfish, the virus is going to need a certain cohort that has not been previously exposed in order to spread efficiently. And usually that happens in the young kids that have been born since the last wave, which is why historically you see viruses circulating the world as pediatric diseases once they've been established, if that makes sense. Yeah. If that makes sense, you get your PhD in epidemiology right now.
Yeah. Well, Dr. Robert Malone, we're out of time. We're going to leave it there, but any final thoughts as we wrap up here?
Only that, stay with it. Use your own mind, make your own decisions. I don't think the government owns us yet, and we still have the right to decide about our bodies, but apparently that's in negotiation right now, and we may all have a vaccine mandate soon. So stay tuned. I have no idea what tomorrow is going to bring.
And I want people to check out your website, rwmalonemd.com.
Thanks. And also, I try to keep the Twitter feed up to date. There are others that are trying to mimic that Twitter feed. But if you look for RW Malone at RW Malone MD on Twitter, and there's about 150,000 followers, that would be the right one.
Sounds good. Thanks for joining us.
Thank you very much for the chance. Hope you feel better.
Thank you.